वेलकम बैक टू अनदर वीडियो। ये वीडियो में हम लोग नीट में जो पहले क्वेश्चंस आ रखे हैं बायोटेक्नोलॉजी के प्रीवियस ईयर क्वेश्चंस वो सॉल्व करने वाले हैं। मैं क्वेश्चन पढ़ने के बाद थोड़ा टाइम पॉज हो जाऊंगा, तो आप उतने में अटेम्प्ट कीजिए, आपका आंसर देने की कोशिश कीजिए। उसके बाद मैं आंसर बताऊंगा, तो देखिए कितना सही हो रहा है आपका और आपका प्रिपरेशन किस लेवल पे चल रहा है। तो पहला क्वेश्चन, विच ऑफ द फॉलोइंग इज नॉट अ क्लोनिंग वेक्टर? YSC, PVR322, प्रोब, BSC। तो इसका सही आंसर है प्रोब। नेक्स्ट क्वेश्चन, जो कि assertion रीजन है। assertion दिया है, when a particular restriction enzyme cuts strand of DNA, overhanging stretches or sticky ends are formed। इसका रीजन दिया है, some restriction enzymes cut the strand of DNA a little away from the center of palindromic site। तो इसका सही आंसर है, both assertion and reason are correct and reason is the correct explanation of assertion। Next question, the enzyme A is needed for isolating genetic material from plant cells and enzyme. Number B, isolating genetic material from fungus। Choose the correct pair of options from the following। ऑप्शंस दिए, सेलुलेज लाइपेज, सेलुलेज प्रोटीएज, सेलुलेज काइटिनेज एंड काइटिनेज लाइपेज। तो इसका सही आंसर है ऑप्शन नंबर सी, सेलुलेज एंड काइटिनेज। नेक्स्ट क्वेश्चन, ligation of foreign DNA at which of the following site will result in loss of tetracycline resistance of PBR322। Options, PST1, PVU1, EcoR1, BAM-H1। तो इसका सही आंसर है ऑप्शन नंबर डी, BAM-H1। नेक्स्ट क्वेश्चन, which of the following methods is not commonly used for introducing foreign DNA into the plant cell? Option A, bacteriophages, agrobacterium mediated transformation, gene gun और disarmed pathogen vectors। तो इसका सही आंसर है बैक्टीरियोफाजेस। नेक्स्ट क्वेश्चन, refer to the following statements for agarose gel electrophoresis। Option A, agarose is a natural polymer obtained from seaweed। The separation of DNA molecules in agarose gel electrophoresis depends on the size of DNA। The DNA migrates from negatively charged electrode to the positively charged electrode। The DNA migrates from positively charged electrode to the negatively charged electrode। तो इसका सही आंसर है ऑप्शन नंबर सी। नेक्स्ट क्वेश्चन, जो कि फिर से एक assertion रीजन है। assertion दिया है, polymerase chain reaction is used in DNA amplification। रीजन दिया है, ampicillin resistant gene is used as a selectable marker to check transformation। तो इसका सही आंसर है ऑप्शन नंबर बी, both assertion and reason are correct, but reason is not the correct explanation of assertion। Next question, which one of the following statement is not true regarding gel electrophoresis technique? Option A, the process of extraction of separated DNA strands from gel is called elution। Separated DNA fragments are stained by using ethidium bromide। The presence of chromogenic substrate gives blue colored DNA bands on the gel। Bright orange colored bands of DNA can be observed in the gel when exposed to UV light। तो इसका सही आंसर है ऑप्शन नंबर सी, the presence of chromogenic substrate gives blue colored DNA bands on the gel। Next question, statement 1, restriction endonucleases recognize specific sequence to cut DNA known as palindromic nucleotide sequences। Statement 2, restriction endonucleases cut the DNA strand a little away from the center of the palindromic site। तो इसका सही आंसर है both statement 1 and statement 2 are correct। Next question, which of the following is not a desirable feature of a cloning vector? Presence of origin of replication, presence of a marker gene, presence of single restriction enzyme site, presence of two or more recognition sites। तो इसका सही आंसर है ऑप्शन नंबर डी, प्रेजेंस ऑफ टू और मोर रिकॉग्निशन साइट्स। नेक्स्ट क्वेश्चन, बिफोर एग्जामिनिंग द बायोलॉजिकल basis of the hereditary human illness sickle cell anemia, a researcher first isolates DNA from an affected person's cell। The subsequent step involves using option A, restriction enzymes which fragment DNA into smaller pieces, at known places. Gel electrophoresis, which isolates the DNA from the other cell fragments. DNA ligases that help DNA bind to one another, or DNA polymerase, which creates new copies of DNA. तो इसका सही आंसर है ऑप्शन नंबर ए, restriction enzymes which fragment DNA into smaller pieces at known places. Next question. The usage of agarose derived from seaweeds is spectrophotometry, gel electrophoresis, PCR and tissue culture. तो इसका सही आंसर है ऑप्शन नंबर बी, जेल इलेक्ट्रोफोरेसिस. नेक्स्ट क्वेश्चन. Plant resistant to dash have been created with the application of RNA interference. Option A, pests, yeast, nematodes and viruses. तो इसका सही आंसर है ऑप्शन नंबर सी, nematodes. Next question. A successful polymerase chain reaction cannot be carried out without which of the following? All four DNA bases, DNA polymerase, both A and B, only A. तो इसका सही आंसर है ऑप्शन नंबर सी, बोथ ए एंड बी. नेक्स्ट क्वेश्चन. Why is that species as different as bacteria and humans can have their DNA analyzed using the same fundamental methods? Every cell in every creature is identical. Every organism has the same quantity of DNA. All creatures have the same DNA sequence. All organisms have the same DNA structure. तो इसका सही आंसर है ऑप्शन नंबर डी, all organisms have the same DNA structure. Next question. What is the process for introducing recombinant DNA into a host cell? Growing the host cell in an ampicillin containing growth media, covering the DNA with carbohydrates to encourage cell engulfment of the DNA, stimulating cells with electrical pulses or calcium ions to make them more permeable, administering proteins to host cells to increase their permeability. तो इसका सही आंसर है ऑप्शन नंबर सी. नेक्स्ट क्वेश्चन. What is an illustration of the gene therapy? The manufacture of the injectable hepatitis B vaccine, the development of vaccination for edible crops like potatoes, the insertion of the adenosine deaminase gene in SCID patients, creating children in test tubes by artificial insemination and implantation of fertilized eggs. तो इसका सही आंसर है ऑप्शन नंबर सी, the insertion of adenosine deaminase gene in SCID patients. Next question. What enzyme is responsible for the removal of nucleotides from DNA's ends? Endonuclease, exonuclease, DNA ligase, N2. तो इसका सही आंसर है ऑप्शन बी, exonuclease. Next question. Which of the following components does biotechnology include? Creation of a gene, in vitro fertilization, fixing a gene that is incorrect, creating a DNA vaccine. तो इसका सही आंसर है सारे के सारे, क्योंकि बायोटेक्नोलॉजी की मदद से हम लोग ये सारे चीज कर सकते हैं.